Hello everybody, and welcome back to biosc140 Human Physiology. This video is going to cover metabolism lectures. Alright, so I'm going to break this video into two parts. The first time through, we're going to cover glycolysis, activation, Krebs cycle, and oxidative phosphorylation. In the second video, we're going to cover all of these, plus we're going to add in a bunch of extra material that is related to different parts of this. This lecture is one of the hardest for the whole semester. I want to give you a heads up. We're going to get through this together, but this section takes effort. It takes repetition. It takes repetition, and it takes compartmentalization of the information. The best way to study this material and to learn this material is to not look at it like a whole. Break it into tiny pieces, study those tiny pieces, and then learn how those tiny pieces, Moose says good luck. Say good luck in studying. Say good luck. All right, bye, Moose. All right, so you have Moose's good luck. It's time to now learn metabolism. It's time to learn how ATP is produced. So compartmentalize. The first compartment that we're going to learn is glycolysis. The first compartment we're going to learn is glycolysis. So glycolysis. Learn this compartment. Glycolysis means sugar splitting. So glyco is sugar, lysis means to cleave or to split, to cut in half. So what are we going to do if in glycolysis? All right, then the word means splitting sugar, means cleaving sugar. We're going to break sugar in half. That's what we do in glycolysis. I want you to know where glycolysis happens. It takes place in the cytosol. It takes place outside the mitochondria. So we're going to split sugar. We're going to cut a sugar molecule in half outside the mitochondria in the cytosol. Glucose has 6 carbons. Glucose has 6 carbons. The whole process of glycolysis is 10 reactions long. You don't need to know the 10 reactions. Once those 10 reactions are done, we have 2 pyruvates. Each pyruvate has 3 carbons in it. Now what's 2 times 3? It's 6. So we start with 6 carbons. We finish with 6 carbons. We start with 1 6-carbon glucose. We finish with 2 3-carbon pyruvates. I want you to know the name of this. I want you to know where it happens. I want you to know what you have at the beginning of glycolysis and what you have at the end of glycolysis. And I want you to know the carbons. Tracking the carbons is going to help you. So glycolysis, you can see here, we start with a 6-carbon glucose. To get the process started, we're going to spend 2 ATP. We're going to use 2 ATP. So you can think of this like, you got to spend money to make money. If you're going to open a business, if you want to make some money, you got to spend some money, right? So we start with a 6-carbon glucose. We want to get as much ATP out of it as we can. And to get started, we got to spend some money. We spend 2 ATP to get the process started. I want you to know that. And we split that 6-carbon glucose into 2 pyruvates, each with 3 carbons. Now, look at this. We gain 2 ATPs per pyruvate. We gain 2 ATPs per pyruvate. So we spend 2 ATP, we earn 4 ATP. So we gain a total of 2. Spend 2, make 4. profit 2 ATP. So look up here at the equation. I want you to know this equation. 1 glucose plus 2 ATP, that's what goes into glycolysis. 2 pyruvate, 4 ATP, 2 NAD is what you get out of it. Sorry, not NAD. 1 second. I got all these windows on my screen. So 2 NADH is what we get out of it. 2 NADH is what we get out of it. Now, the NADH is important. You're going to learn all about why it's important later on. But NADH is a molecule that carries a lot of high energy electrons. You can see NADH here. You can see NADH here. It carries a lot of high energy electrons. And we're going to use those high energy electrons at a later date to cash in more ATP. So that ATP is going to be made at a later date. We'll get more on that later. There's one other thing I want you to notice here. This molecule NAD+ gains those high energy electrons and becomes NADH. NAD+ and NADH are kind of like ATP and ADP. It's like they go back and forth between each other. NAD+ lacking high energy electrons. NADH has high energy electrons, and it kind of goes back and forth. So both of these are going to be important later on. So remember those. All right, so I'm going to skip over a number of slides the first time through. The second time through, we're going to touch on all of these slides that I'm skipping. But right now, I want you to get the glycolysis compartment set. I want you to get the activation compartment set, the Krebs cycle compartment set, and the oxidative phosphorylation compartment set. On the second video, we're going to go through and we're going to add in a bunch of extra things to it and see how they all connect. So, glycolysis, check. We've learned about glycolysis. Now, let's move on. So, once we have our glycolysis, everything we just went over, put that in a box. That's the glycolysis box. Now, once we have our glycolysis box, we can either go left or right. There's two directions that we can go after glycolysis. We can either go anaerobic or we can go aerobic. So at this point, we haven't talked about oxygen. So glycolysis happens with oxygen or without oxygen. What happens to the pyruvates is different if we do anaerobic respiration or aerobic respiration. So I'm going to repeat that. We have not talked about, or when we talked about glycolysis, we did not talk about oxygen. Glycolysis is going to happen up to this point, just like we said, in the presence of oxygen and without oxygen, same way. What's going to happen to the pyruvates will vary depending on if we go the anaerobic pathway or the aerobic pathway. So this is our first split. Are we going left? Are we going right? Are we going anaerobic or aerobic? We're going to look over anaerobic first. So now we're going to look at the fate of the glycolysis products in an anaerobic situation. So anaerobic, what we're going to do, well, think about it like this. We profited 2 ATP during glycolysis. We're left with this NADH and this pyruvate. The process of glycolysis requires that we have an NAD+, that 2 NAD+. Well, we just turned our NAD+ into NADH. So we need to turn our NADH back into NAD in order to help the glycolysis, in order to allow more glycolysis to happen. So glycolysis happened, we have NADH, we have pyruvate. We need to turn NADH back into NAD+ in order to do more glycolysis. So we combine pyruvate and NADH, and we get lactate and NAD+. You've probably heard of lactic acid. Right here, lactic acid. So in order to continue to do glycolysis, we combine NADH with pyruvate. The NADH turns back into NAD+, which can help do glycolysis more. The pyruvate turns into lactic acid. Lactic acid is going to be, we'll talk about what lactic acid goes and does later, but lactic acid will go into your bloodstream, it'll leave the area. This cell right here, lactic acid will leave the area. We'll talk about what it does when it leaves the area later on the second time through this lecture. Right now though, just know anaerobic respiration, we combine NADH with pyruvate, we get NAD+ again, more glycolysis happens, and the pyruvate turns into a 3-carbon lactate. Anaerobic respiration regenerates NAD+ so that glycolysis can continue. It's a small but rapid yield of ATP. Remember, we spent 2, we made 4, we profited 2. No O2 requirements. No O2 requirements. Now, I guess this is a little flavor on top, but at any moment, all of your cells are doing anaerobic and aerobic respiration. Sometimes they might have that ratio might change where a cell's doing more anaerobic than aerobic, but human cells are always doing both, anaerobic and aerobic. All right, so now we have our glycolysis, and then we go left or right. We go anaerobic or aerobic. We just covered anaerobic. Anaerobic glycolysis, right here is the summary chart. I want you to know these summary charts. We used 2 ATP, we profited 4, or we used 2 ATP, we got 4 ATP out of it. Total ATP produced 4, we profited, we have a net profit of 2 ATP. All right, we're now gonna go to our next aerobic respiration. So now we're going the other direction. We're going aerobic after glycolysis. Aerobic, as the name implies, will use oxygen. So we just did glycolysis. Now we're going down the aerobic pathway. The aerobic pathway is going to take us to pyruvate activation, which is our next little compartment, our next box, which is going to lead to Krebs cycle, the following box, which is going to lead to electron transport chain, which is our final box. So let's move right along to pyruvate. All right, so right here's like glycolysis. We've already gone over that. Now let's move to our next. So we're going the aerobic direction. We're going to pyruvate activation. So glucose happens in the cytosol. Under aerobic conditions, we're going to take those pyruvates and those NADHs, and we need to move them into the mitochondria to cash in on more ATP, more energy from those products. So when we move pyruvate into the mitochondria, this process is called activation. And we're going to have 2 pyruvates moving in. They're going to interact with 2 NAD+. They're going to interact with 2 NAD+. We're going to, so input is 2 pyruvates, 2 pyruvates. Let me take a step back. We're following the journey of 1 glucose molecule. You can activate 1 pyruvate at a time. These equations and this story that I'm telling is the story of 1 glucose. Because 1 glucose splits into 2 pyruvates, the story is going to include 2 pyruvates as we move forward. But, you know, really each pyruvate is going to do this individually. Each pyruvate is going to go in individually. We could talk about 1 individual pyruvate. We're talking about 2 because we want to start with 1 glucose and follow all that glucose through the whole story. So 2 pyruvates move into the mitochondria. When they move into the mitochondria, we're going to lose 1 carbon, and that carbon is going to be released as carbon dioxide. So in this class, we've already talked about how CO2 is a byproduct of energy production. Right here is one of those CO2s. It's going to be 1 CO2 per pyruvate. So we start off with 3 carbon pyruvate. You can see right here in gray, we have a carbon and 2 oxygens, a CO2. We're going to lose 1 CO2 per pyruvate, leaving us with something called acetyl-CoA. Acetyl-CoA has 2 carbons. So we start with 3 carbon pyruvate, we end up with 2 carbon acetyl-CoA. We also turn a couple NAD pluses into NADHs. And remember, NADHs are molecules that carry a lot of electrons, like high energy electrons, and we're going to use those to produce more energy, more ATP later on. So, pyruvate, you gotta activate it, you gotta move it into mitochondria. When you move it into mitochondria, 1 carbon dioxide comes off per pyruvate. You also turn 1 NAD plus into NADH per pyruvate, leaving you with an acetyl-CoA, 2 carbons. Acetyl-CoA is going to be the input into Krebs cycle. Krebs cycle is our next compartment. So now we move on to our next compartment. We take our acetyl-CoA, which was the product of activation, and we're going to put our 2 carbon acetyl-CoA into Krebs cycle. Krebs cycle is a cycle. It goes around in a circle. We're going to add in that acetyl-CoA that we made, and we're going to get 1 turn of the Krebs cycle. We're going to add in acetyl-CoA, we're going to get 1 turn of the Krebs cycle. We're going to add in acetyl-CoA, we're going to get 1 turn of the Krebs cycle. Because we're talking about the journey of 1 glucose molecule, which splits into 2 pyruvate molecules, which turns into 2 acetyl-CoA molecules, these numbers down here are for 2 circles of the Krebs cycle. We could talk about 1 acetyl-CoA going into Krebs cycle, be 1 spin. These numbers down here are for 2 turns because of the reasons I just stated. A little heads up. So the scientist Krebs, he discovered a lot of things. And the name Krebs cycle is kind of going out of style, because when you say the Krebs cycle, it's kind of like, well, what cycle? Because, you know, Dr. Krebs discovered like a bunch of different things in physiology, a bunch of different cycles. So Krebs cycle, for that reason, has fallen out of favor, and it's being called pretty often the citric acid cycle now. So you'll hear it called Krebs cycle and citric acid cycle. It's the same thing. So we put in our 2 carbon acetyl-CoA. It goes around. Now, for every 2 acetyl-CoAs that we put in, we turn 6 NAD pluses into 6 NADHs. That's a lot of high energy electrons. We turn 2 FADs into 2 FAD pluses. FAD and FADH2 are very similar to NAD plus and NADH. They're high energy electron carriers that we're going to use an electron transport chain to make more ATP. And we get 2 ATP directly in this cycle. So this is for 2 acetyl-CoAs. We put in 2 acetyl-CoAs, we do 2 turns, we get 6 NADHs, 2 FADH2s, and 2 ATPs. 1 ATP per cycle, 1 ADH per cycle, 3 NADHs per cycle. I also want you to pay attention to the carbons. For every turn, you lose 2 carbons to carbon dioxide. So acetyl-CoA has 2 carbons. You put 2 carbon acetyl-CoA in. During that spin, 2 carbons are lost to carbon dioxide. You put 1 more acetyl-CoA in, which has 2 carbons. You do the spin, you lose 2 CO2s there. So you put in 2 acetyl-CoAs, you lose 4 CO2s. Now, we have lost all 6. At this point, we've lost all 6 of our carbons from the original glucose to carbon dioxides. For every glucose molecule that goes down this aerobic respiration, this aerobic metabolism pathway, every one of those carbons is going to get turned into a different CO2 molecule. So it's probably making a little more sense now why in our first few labs we saw that energy usage produces so much CO2. It's right here. That's why. All right, so at this point, we profited 2 ATP in glycolysis. We profited 2 ATP here in Krebs cycle or citric acid cycle. That's not a lot of ATP. That's really not a lot of ATP. So we're going to get most of our ATP from this final compartment, this final process. In this final process, we're going to take all these NADHs and FADH2s that we've made up to this point, and we're going to cash in on a ton of ATP. So right here, with glycolysis, we get a couple ATP. We do pyruvate activation, we do citric acid cycle, we get a couple ATP. We're going to take NADHs from glycolysis. We're going to take the NADH from pyruvate activation. We're going to take the NADH and FADH2s from citric acid cycle, and that's what we're going to be inputting to the electron transport chain, to this oxidative phosphorylation, I'll talk about that later, on this electron transport chain, where we're going to get tons of ATP. So let's look at this last box. All right, so electron transport chain. We are in the mitochondria at this point. Remember, Krebs cycle, citric acid cycle takes place in the mitochondria. We move our pyruvate from glycolysis into the mitochondria. So we're in the mitochondria for activation. We're in the mitochondria for citric acid cycle. We're in the mitochondria for electron transport chain. Mitochondria, you need to know something about the anatomy of a mitochondria. It's a bimembrane system. So we have an inner membrane and an outer membrane. Yes, every mitochondria has 2 phospholipid bilayers. It has an inner one, an inner membrane, and an outer membrane. What we're going to do is we're going to use those NADHs and those FADH2s, those high energy electrons there. We're going to move hydrogen ions into the intermembranous space. We're going to move hydrogen ions from the mitochondrial matrix, the inside of both the inner chain, most inner chamber of the mitochondria. We're going to move hydrogens into the intermembranous space, the space in between the 2 membranes. So we're going to have a high concentration of hydrogen ions in this intermembranous space. And what do things like to do when there's concentration gradients? Things like to go down concentration gradients. So these hydrogen ions are going to have high concentration in between the membranes, lower concentration within the most inner membrane. Those hydrogen ions are going to flow down their gradient through an enzyme called ATP synthase. Yes, ATP synthase synthesizes ATP. Those hydrogen ions are going to flow through this molecule, this protein, and as they flow through this protein down their concentration gradient, they are going to power the process of putting a phosphate group onto ADP. When you put a phosphate group onto ADP, you get ATP. So electrons transferred to electron transport chain, energy released during transfers hydrogen ions into the intermembranous space. Chemiosmosis. So chemiosmosis it describes the hydrogen ions flowing from this intermembranous space through ATP synthase into the inner mitochondrial matrix and producing ATP. Oxygen is required for this process and is the ultimate electron acceptor. So oxygen sucks those high energy electrons through this process. There's this electron chain, and the place where those electrons end up is on oxygen. And when those electrons end up on oxygen, it turns an O2 into H2O. I'll repeat that. Those high energy electrons get sucked into this protein transport, this protein system that moves the hydrogens across the inner membrane. Those electrons flow through these proteins, and the final thing that's pulling those electrons through this process that finally captures those electrons is oxygen. And when oxygen captures those electrons, it's going to form water. It's going to combine with some hydrogen ions, it's going to form water. So oxygen is the ultimate electron acceptor. All right, so a few more things. NADH, for every NADH you have, it produces about 3 ATP. For every NADH you have going into this, you get about 3 ATP. For every FADH2 that you have going into this, you get about 2 ATP. Remember those numbers, they're important. All right, so let's look at the summary charts. Glycolysis, sugar splitting, aerobic. So the aerobic situation. We put in the glucose, we use 2 ATP. We get 4 ATP direct. Direct means you get the ATP right then and there. So we get 4 direct in glycolysis. We produce 2 NADHs in glycolysis. We produce 0 FADHs in glycolysis. The indirect ATP is the ATP we get from NADH. I'm going to say this again because this is a confusing part of this. These charts are great for studying, they're great for understanding this whole thing, but the one thing that throws people off is this idea of indirect ATP. Indirect ATP is ATP that is produced from NADH or FADH2 in the electron transport chain. Direct ATP is produced right then and there in glycolysis or right then and there in Krebs cycle. That's how these charts work. All right, so we produce 2 NADHs in glycolysis. So what's 2 times 3? Because we get 3 ATP per NADH. Well, it's 6. So 6 indirect ATP, 4 direct ATP per glucose. 4 plus 6 is a total of 10 ATP. Now we spent 2 ATP to get the process going, so 10 minus 2, we profit 8 ATP in aerobic glycolysis. Now, what about during pyruvate activation? So in pyruvate activation, we don't get any direct ATP, but we get 2 NADH. We get 3 ATP per NADH, so 3 times 2 is 6 indirect. 6 indirect plus 0 direct, 6 total, 6 net. And we lose 2 carbons to carbon dioxide. Krebs cycle, we get 2 direct ATP. We get 1 ATP per spin of the Krebs cycle right then and there. We produce 6 NADHs for our 2 cycles. We produce 2 FADHs. 6 times 3 is 18. 2 times 2, because we get 2 ATP per FADH2 is 4. 18 plus 4, 22 indirect ATP, plus the 2 direct ATP that we get, 24 ATP total, 24 net. That's how you read these graphs. Know these graphs. I know these graphs for the test. Study them. All right, and here is the summary chart of it all. Here's a summary chart of it all for the complete oxidation of a glucose. I'm going to stop this video here. What I recommend is you spend you spend more time going through your notes for this lecture. Maybe even watch this lecture again. Tomorrow, watch this lecture again, go over your notes again. And then at that point, after you've gone over it a couple times, proceed to the next video. This topic takes repetition. This topic takes compartmentalization. Just learning glucose Easy, you can totally do it. Just learning activation, easy, totally can do it. Just learning Krebs, just learning electron transport, totally doable. Once you've learned them, put them together, see how they interact. If you look at it as a whole big picture, like one big box, it gets really tough to study. So study the boxes, see how the boxes interact. I'll see you in the next video. Good luck studying.